Greetings of the day. I am Dr. P. Saujanya. I welcome you all to the lecture on growth of bacteria. In this lecture, I will describe different methods of reproduction of bacteria and how the bacterial growth has been measured. So when bacteria are inoculated in a suitable medium and maintaining an appropriate conditions, a tremendous growth or increase in number of bacterial cells will occur within a short period of time. And this time period varies according to the type of the bacteria. And determination of growth, it requires quantitative measurement of total population of the cells at the time of inoculation and again after incubation. So the number of bacterial cells inoculated at the starting of the inoculation and after incubation, how many cells has been produced will give the determination of growth of bacteria. Usually, bacteria will reproduce by transverse binary fission process. And this binary fission process has been occurred by the development of transverse septum in the cell. So here, one cell will divide into two cells will divide into four. Likewise, in the geometric progression manner, the cells will divide. These are the general methods of cell division that occurs in bacteria. Basically, the cell division is by binary fission process or by budding process, fragmentation, or by the formation of pores. So in the first image, you can see the bacterial cell size has been enlarged and the development of the septum. And this development of the septum divides the cell into two and it produces two daughter cells. And this is an example of transverse binary fission in Bacillus subtilis. And similarly, this is the example of transverse binary fission in Streptococcus faecalis organism. This is a cocci shape and this is a rod shaped bacteria. And another example is transverse binary fission in prosticate bacteria, that is Prosticobacter fusiformis. So here you can see the formation of the septum in the prosticate bacteria, and this septum divides a single bacterial cell into two daughter cells. And the next method of cell division is by budding. And a small bud has been developed at one end of the bacterial cell, and this bud will gradually develop into an individual cell and it develops as a daughter cell. So this is an example of budding in Rhodopseudomonas acidophila. Next, budding example in Hyphomicrobium vulgare. And then fragmentation. Here the bacterial cell has been enlarged and different septums has been formed in the bacterial cell, and each fragment will develop into an individual bacterial cell or daughter cells. And this is the method of cell division in Nocardia, fragmentation process in Nocardia species. And lastly, this is an example of spore forming bacteria. And here the formation of conidiospores has been observed in Streptomyces species. This is the conidiospores formed in Streptomyces species. These are the various modes of the cell division that occurs in bacteria. So generally the bacteria will reproduce by binary fission process. And this binary fission process is an asexual reproduction. And here the cell increases in size and the cell components has been doubled. The cell size has been increased and the cell components has been doubled. And here one cell will divide into two and those two cells will divide into four and four cells will divide into eight, and likewise the cell number has been increased exponentially. And this cell division has been formed by the formation of septum in between the cell. So the microbial growth is defined as an increase in number of cells in the population. And the time taken for the population to get double is known as number of generation. That is generation time. The time required to get to double the population is generation time or doubling time. So whatever the septum that has been formed, it projects inward and then it makes the division of the one bacterial cell into two daughter cells. The two daughter cells has been separated. And here the cell division varies according to the species of the organism and the conditions which has been maintained during the incubation or during growth of the organism. For example, if we take E. coli, Escherichia coli organism, it can double in 20 minutes. Within 20 minutes, the bacterial cell will double. And in case of mycobacterium, it requires at least 12 hours to get the double. So this bacterial growth has been measured and it is studied as bacterial growth curve. When an organism has been inoculated in the medium, so the cell division occurs in geometric progression manner. That is, one cell will divide into two, two cells will divide into four, and four cells will divide into eight, likewise in geometric progression manner. That is 2 power 2, 2 power 3, 2 power 4, 2 power 5, 2 power 6, likewise till 2 power n. So here n is represented as number of generations, and this has been calculated based on the number of the organisms inoculated in the medium. So assuming that there is a no cell death during the growth of the organism, so n is uh, calculated using the formula 1 into 2 power n. N is the total population. The total population that has been formed during the growth of the bacteria in the medium. That is n. The total population is equal to 1 into 2 power n. 1 is initial population considering inoculated one organism in the medium. 2 power n. 2 power n is number of generations. So, as it is highly impossible to inoculate a single organism, this one has been replaced with N naught. So, N naught is initial population. So, if you take an organism, a loopful of organism from a medium, then it contains multiple number of organisms, thousands or millions, trillions of organisms. So, it is highly impossible to pick a single organism from the culture. That is why one has been replaced with N naught. N naught is initial population. So, the formula has been written as N is equal to N is total population is equal to initial population into 2 power n. So now apply the logarithms on both sides. So log base 10 n, that is log of total population, is equal to log base 10 n naught, that is log of initial population, plus n log base 10 2. So here n, number of generations, has been calculated using the formula log base 10 n, that is log of total population minus log base 10 n naught, that is log of initial population divided by log base 10 2. And here replace the value log base 10 2, that is 0.301. So n is equal to log of total population minus log of initial population divided by 0.301. So 1 by 0.301 is 1 by 0.301 is equal to 3.3. So the number of generations n is equal to 3.3 into log of total population minus log of initial population. Using this formula, we can calculate number of generations, and based on the number of generations, how many cells has been produced, we can calculate. So when the organism has been inoculated in the medium, this organism undergoes into various phases, and that has been studied in bacterial growth curve. So the bacterial growth curve indicates 4 stages. That means the bacteria will undergo in 4 stages when the organism is inoculated, and after incubation till death it undergoes 4 phases. They are: the first phase lag phase, next second phase log phase, and then third phase stationary phase, and the fourth phase is death phase. So here the first phase lag phase is when the organism is inoculated in the medium, immediately the organism cannot grow. So it takes certain period of time to adjust to the medium. That is intense activity preparing for population growth. So it starts the production of enzymes and coenzymes, and there is no cell growth or no population, no increase in population in the initial stage, that is lag phase. So once it adjusts to the new environment and it starts utilizing the nutrients, so once it starts utilizing the nutrients, the metabolic activity of the organism is tremendously increased. That is, that phase is denoted as log phase or exponential phase. So the second phase is logarithmic or exponential phase, and increase in population. So here the number of cells dividing at the population is more when compared to the number of cells dying. And then if you are not transferring this organism into the fresh medium and the same medium has been maintained, then what happens? The organisms whatever already has been grown, they starts to produce the byproducts, and these byproducts may be toxic to that of the bacterial growth. So at a certain period of time, if you are not adding the fresh medium to the bacteria, or if you are not transferring the bacteria to another fresh medium, then there occurs a static phase, which is known as stationary phase. So in the stationary phase, the number of organisms growing and the number of organisms not growing, that is that undergoing death, is equal. So there is no new population occurs in the stationary phase. So this phase is also known as period of equilibrium. So the microbial death balance production of new cells. So here it's due to the accumulation of the byproducts. Now with the release of the toxic substances, the bacteria cells will start dying. So here there is no new cell growth population. Even though if the organisms are not provided with the fresh nutrients, or they are not transferred to the fresh medium, then due to the accumulation of the more toxic substances or byproducts, the cells will undergo death phase. So all the cells will undergo death phase. The population is decreasing at a logarithmic rate. So it is quite opposite to that of the second phase. Second phase, rapid growth, exponential growth occurs. So due to the high nutrient level. And in the fourth phase, due to depletion in the nutrient level, and that logarithmic rate of decrease in population, that is death phase occurs. That is the death phase. So the bacterial growth curve has been plotted when the bacteria are inoculated into a liquid medium, and we can plot the number of cells in the population over a period of incubation. And 4 phases of bacterial growth, and the first phase is lag phase. This is the period of adjustment to new conditions, and the bacteria will adjust to the new environment, and there is a little or no cell division occurs, and the population size doesn't increase here, it is stagnant. And the phase of intense metabolic activity in which individual organisms grow in size, and this may last for 1 hour to several days. This lag phase depends upon the generation time, the time required for the bacteria to get the double. So it lasts for 1 hour to several days. So next, the bacteria after starting utilizing the nutrients and adjusting to the new environment, it undergoes log phase. That is, the cells divide rapidly and the generation time reaches a constant minimum. And this is a period of rapid growth. Here, the number of cells produced are more than number of cells dying. So considering there is no cell death, all the cells are in growing stage. And the cells are at high metabolic activity in log phase. And these cells, when they are growing in the medium, they may be susceptible to that of certain environmental factors like radiation and antibiotics. If the cell growth is exposed to radiation or any antibiotics, then that leads to the death of the cells. Next, stationary phase. Stationary phase here, the population size begins to stabilize and the number of cells produced is equal to number of cells dying. And overall here, the cell number does not increase and the cell division begins to slow down. The cell division will begins to slow down. And the factors which influence the cell slowdown of microbial growth are due to the accumulation of toxic waste materials are due to change in the pH, that is acidic pH of the medium, because due to the release of all byproducts, the pH of the medium may be decreased due to acidic condition. The acidic pH is toxic to the growth of bacteria. Are limited nutrients. So as you are not adding the fresh nutrients to the medium due to the uh, due to the depletion in level of the nutrients, also the cell death occurs. And insufficient oxygen supply also may lead to the cell death. These are the factors that affects the microbial growth, slowing the microbial growth during stationary phase. And the death phase or decline phase. Here, the population size begins to decrease in the population size. And here the number of cells dying is more than number of cells produced. This is quite opposite to that of log phase. And the number of cell number decreases at logarithmic rate, and cell lost their ability to divide and few cells may remain alive for a long period of time. And next, synchronous growth. Synchronous growth is nothing but maintaining the organism in a single phase. In the bacterial growth curve, when the organism is inoculated in the medium, it undergoes various phases. So if we take a sample of organism, the sample contains the organism which are in mixed phases. And to maintain the organism in a single phase, synchronous growth is used. So here, a population can be synchronized by manipulating the physical environment or the chemical composition of the medium. Either way, you can manipulate the physical environment or chemical composition of the medium. The synchronous growth can be obtained. So for example, if the cells are inoculated in a medium at suboptimal temperature, and if they are kept in this condition for some time, and they will metabolize slowly, but they will not divide. Suboptimal temperature. Usually, when the bacterial cells are inoculated in the medium, they should be incubated at room temperature. If you are incubating at a lower temperature, so what happens? All the cells will be in dormant stage, and the metabolic activity at the low temperature is very slow. So when the temperature is subsequently raised, when you have been incubated or transferred that inoculated cells to the room temperature or rising the temperature, what happens? The cells will undergo a synchronized division. That means all the cells would have been adjusted to the new environment and they start utilizing the nutrients, and their cell growth is rapid and cell division is rapid. Likewise, so once again, if you inoculate the organism in suboptimal conditions, again the metabolic activity will be lowered and the cell division is reduced. Subsequently, if again you have been raised the temperature, then the cell division is synchronized. In this graph, you can see. So initially, if you inoculate the organism and incubate at suboptimal condition, so the growth of the organism, suboptimal condition, and then transferred to the optimal temperature condition, then the growth of the organism is rapid. Once you have been inoculated, once you have been incubated at a suboptimal condition, the growth has been stagnant. There is no growth. And once you have been transferred again to the optimal temperature condition, the rapid growth occurs. So alternatively, if we are changing the physical environmental condition of incubation of the medium, then you can obtain the synchronous growth of the bacteria. Next, continuous culture. Till now, we have discussed the batch culture, inoculating the organism in the medium and incubating for certain period of time. In continuous culture, you can grow the organisms continuously by providing the fresh nutrients. So, these continuous culture methods includes two types, chemostat and turbidostat. Chemostat is here the concentration of an essential nutrient, that is the substrate, within the culture vessel will be controlled by the growth rate of the cells. So here it is controlled by the dilution rate. Dilution rate is, that is the rate at which fresh medium is being added to the culture divided by the volume of the culture vessel. The rate at which fresh medium is being added to the culture divided by the volume of the culture vessel. So therefore, by adjusting the dilution rate, the growth is controlled. For example, if the dilution rate is low, so here in this image, you can see this is a culture vessel and this is the reservoir that containing the medium, and the culture vessel has been supplied with the air, and it also contains a port for the inoculum. And whatever the spent medium that is present in the culture vessel is transferred to the spent medium vessel or receptacle. So in this medium, once you have been inoculated the organism and maintained optimum conditions, then the organism will grow. So this dilution rate has, if the dilution rate is increased or if the dilution rate is low, that means if you are slowly adding or if you are after certain period of time, if you are adding the medium to the culture vessel, that means uh, the cells has been given a certain period of time to use the nutrients properly and grow well. So what happens if the dilution rate is low? The cell reaches high density. If you are giving a certain period of time to grow the organism, utilizing the nutrients, the cell density increases. That means you are not eliminating the, you are not removing the culture into the receptacle periodically. Similarly, if the dilution rate is high, that means you are not giving a certain period of time to the culture that is present in the culture vessel to use the nutrients and grow. If the dilution rate is high, once you are adding the medium to the culture vessel and removing the medium already spent in the culture vessel, what happens? If you are giving a less period of time to grow the organism in the medium, what happens? The cell will not grow properly, so the cell density is low. So the dilution rate, if the dilution rate is low, the cell density is high. That means it is given a certain period of time to use the nutrients and grow. If the dilution rate is high, that means you are continuously adding the fresh medium to the culture vessel, what happens? It will not be given the time to use the nutrients, so the cells will not grow properly. That is why the cell density is low. This is chemostat. And the next method is turbidostat. In this turbidostat is measured by photoelectric device. This photoelectric device monitors the turbidity of the culture vessel. Here, this culture vessel turbidity has been controlled by dilution rate to maintain the cell density at a constant rate. If the density becomes too high, the dilution rate is increased. So once you have been measured the turbidity, the cell density indicates here turbidity, that gives turbidity. In a photoelectric device, you will measure the cell density in in uh, inverse of turbidity. So if the cell culture is too turbid, what happens? The cell density that indicates the cell density is too high. So if this cell density is too high, it should be reduced. The cell density has to be reduced by increasing the dilution rate. Increasing dilution rate means adding fresh nutrients to the medium. So thereby the cell density will be decreased. So that means if the cell density is high, that the dilution rate is increased. If the cell density is too low, that means the organisms are not grown properly, then the dilution rate is decreased. That means you should give some period of time to grow the organism. So finally, if the cell density becomes too high, the dilution is increased, and if the cell density becomes too low, the dilution rate is decreased. So these two methods are continuous culture methods. The next topic is measuring microbial growth. How this microbial growth has been measured. So the measuring of microbial growth is by direct method of measurement and indirect method of measurement. In direct method of measurement, there are various methods like plate count. So in the plate count here, the bacterial population has been counted or measured after incubation, after inoculation and incubation. In plate count method, inoculate the organism in a medium containing Petri plate with the sample. The sample is of organism and count the number of colonies. This is the principle involved in plate count method. And the assumptions in plate count method are each colony originates from a single bacterial cell and the original inoculum is homogeneous. The mixture is homogeneous. That means it should not contain multiple organisms and no cell aggregates are present in the sample whatever we have taken for plate count. And the advantages of this plate count are it is used to measure viable cells, and the disadvantages are it takes around 24 hours because after inoculation you need to incubate for 24 hours to develop the visible colonies, and it only counts between 30 to 300 colonies. So we have to choose a plate that is producing 30 to 300 colonies. Below 30, above 300 colonies producing plate is not advisable, and it must perform serial dilutions to produce or to get appropriate number. So whatever the sample is taken for counting the bacteria, that should be diluted by serial dilution process. So this is the serial dilution process here. Based on the number of dilutions, you have to choose the test tubes. Here in this image, 5 test tubes has been taken and each test tube is filled with 9 ml of the sterile broth, nutrient broth. Broth is liquid medium has been filled. In the first tube, 1 ml of the original inoculum or the stock inoculum has been added. So it contains 10 ml, 9 ml of nutrient broth and 1 ml of the inoculum. Total 10 ml. Mix it properly, then transfer 1 ml from first tube to the second tube. Likewise, from second tube to third, third to fourth, fourth to 5, and from the last tube discard 1 ml. So as the original inoculum is diluted from first tube to last fifth tube, the number of organisms present in the medium has been reduced serially. That is serial dilution process. So here the dilution rate is 1:10, 1:100, 1:1000, 1:10,000, and 1:1 lakh dilution. And when you pick the sample from these diluted samples, you can see this plate contained more number of organisms where you are not able to count the organisms. They are not distinguished properly. And then the next second dilution, it also produced clumsy organisms where you cannot count. And the third dilution, this also produced a group of cells. The fourth plate, you can see the organisms which are countable individual cells. So we need to pick the organism. We need to pick the dilution in such a way that the dilution should produce 30 to 300 colonies. Below 30 or above 300 is not considered. In the last plate, you can see the sample water has been plated here. It produced very less organisms. That is also not considered. Below 30 cells or colonies are also not considered. This is the process of serial dilution. So this serially diluted sample, now it has to be inoculated in the medium. So in the plate count method, introduce 1 ml or 0.1 ml of inoculum into an empty Petri plate. Here in the image, you can see 1 ml or 0.1 ml is inoculated. 1 ml of serially diluted sample, and then it is mixed with the agar. Agar medium when it is in liquid medium. At 50 degrees centigrade, it is in liquid medium. Then mix it properly and pour it into the Petri plate. That is, as you are pouring it into the Petri plate, it is known as pour plate method. And gently mix properly, rotate the plate so that the sample has been mixed with the medium evenly. And the disadvantages of this medium are not useful for heat sensitive organisms. So here you are mixing the sample in the medium at 50 degrees centigrade temperature. So the organisms which cannot withstand at this temperature, they cannot grow here. And here as it is mixed with the medium, the subsurface or under the agar organisms also grow. That is another disadvantage using plate count method. And the next one is spread plate method. In the spread plate method, the inoculum, the serially diluted inoculum has been added on the surface of the Petri plate containing the medium. So this is a Petri plate and the medium has been poured in the Petri plate, and after solidification of the medium, the serially diluted 0.1 ml of bacterial suspension is added. This bacterial suspension now has been spread evenly on the surface of the medium by rotating this sterile glass rod or L rod. So this spreads the bacterial inoculum evenly on the surface of the medium. When this plate is incubated in an incubator for 24 hours, then it produces isolated colonies which are able to count. So the colonies grown on the surface of the medium. That is the advantage using four plate method. In the spread plate in spread plate method. In the pour plate method, both surface and subsurface colonies has been produced. That is, producing subsurface is the disadvantage in pour plate method. In the spread plate method, only the organisms are grown on the surface. And the advantage of spread plate are colonies will grow on the surface and not exposed to the melted agar. And the next method of direct method of measurement of microorganism is filtration. Till now we have been discussed plate count. In plate count, pour plate method and spread plate method are the two techniques. In filtration, this is used to filter a small quantity of the bacteria, and the large sample can be used to filter. So this especially used for filtering the ocean water that is contaminated with fecal bacteria. And a large sample like 100 ml or more can be filtered to retain the bacteria, and the filtrate is transferred onto the petri dish and then incubate and count the colonies. So here you can see this is a filtration assembly. You can see this is a membrane filter. Membrane filter have a very less pore size, around 0.2 micrometer in diameter, which filters the bacteria. Now through this membrane filter, the sample has been filtered, and whatever the bacteria present in the sample will retain on the membrane filter pad. Now this membrane filter pad is inoculated in inward position. That is, the bacteria which are retained on the membrane filter pad should touch to the media. In such a way, it should be inoculated. And after incubation for 24 hours, if this membrane filter pad has been filtered the bacteria, they will grow on the medium. So these are the colonies that has been grown on the medium. So this is membrane filtration method for counting the bacteria. Next, most probable number, MPN, most probable number. This is also a direct method. Here, it is used to measure bacteria that will not grow on the solid medium, which will not grow on the solid medium. And here, the sample, whatever you are counting the colonies, that sample has been inoculated in several broth tubes for each dilution point. So it is serially diluted in broth. It is not in agar. It is diluted in broth medium. And count the number of positive tubes in each set. So when you identify the turbidity of the organism, you can see the growth of the organism. The growth of organism indicates turbidity. So once you have been diluted the sample in the broth and after incubation, so in which dilution the growth is observed, in which dilution the growth is not observed, you can identify by measuring the turbidity. And the statistical method for this most probable number method is, it determines 95% probability that a bacterial population falls within a certain range. The next, direct microscopic count. This direct microscopic count, it requires a special chamber, that is Petrov-Husser's chamber. On Petrov-Husser's chamber, the bacterial culture is added and then a stain to visualize the bacteria, a coloring agent, a stain is also added. And now cover with the cover slip and using the microscope, you can count the number of bacteria in direct microscopic count. And whatever the cells we have been counted under the microscope, they have been multiplied by a factor to obtain the concentration of bacterial cells present in the sample. And the advantages of this method is there is no incubation time required for direct method of measurement. And disadvantages are, you cannot distinguish between live and dead bacteria, and the motile bacteria are difficult to count by direct microscopic count. As they are motile, they will be moving so that you cannot count the bacteria properly. And it requires high concentration of the bacterial sample, that is 10 million cells per ml. So this is a Petrov-Husser's chamber that contains a square for the counting of the bacteria. On the Petrov-Husser's chamber, the bacteria culture sample has been added and the stain has been added. Mix it properly, then cover with a cover slip. And when you observe under the microscope, you can find the bacteria that has been present in the sample, and these bacteria has been counted. Next, indirect method of measurement of bacteria. And the indirect method includes turbidity metric method. So here, the bacterial culture that is grown in the broth medium, their turbidity has been measured using spectrophotometer to determine the percentage transmission or absorbance. And based on the absorbance, the turbidity of the bacterial suspension is calculated. And this is multiplied by the factor. That is, factor is dilution rate. Dilution rate is, for example, 10 power 2, 10 power 3, 10 power minus 2, minus 3, minus 4, like that. That is the dilution factor. Based on the dilution factor, and that a number of cells that has been counted as absorbance, it is multiplied with the dilution factor. And the advantage of this turbidimetric method is no incubation time is required. Directly you can take the culture, then you can measure the turbidity using spectrophotometer. And disadvantages are, cannot distinguish between live and dead bacteria. Both are counted here by turbidimetric method. And it requires high concentration of bacteria, 10 to 100 million cells per ml. Next, electronic enumeration of the bacteria. In electronic enumeration of bacteria, an electronic device like a Coulter current electronic device is used to count the bacteria. In the image, you can see the microbial suspension has been passed through a narrow orifice. This is a narrow orifice. When the microbial suspension has been found in this suspension, microbial cells are found in this suspension, then the electric current that flows through the orifice will be detected, and it is automatically counted as a colony in automatic counter. Okay, so this also counts both the dead and living cells, and this is a very easy method for counting and useful for large microbial suspension volumes also. The next indirect method is metabolic activity. So based on the metabolic activity also, you can measure the microbial growth. As bacteria multiply in the medium, they produce certain products like carbon dioxide and acids as a byproduct. So based on the measurement of acid or the carbon dioxide, you can also detect the metabolic activity of the organism. And this method is very expensive method. And the dry weight. Dry weight also determines the indirect measurement of the microbial growth. Here, bacteria or fungi in the liquid medium are centrifuged. Once the bacterial suspension or fungi suspension has been centrifuged, then all the cells will settle at the bottom as a pellet. And this supernatant liquid has been discarded and the pellet has been weighed. And this pellet weight determines the dry weight of the microbial cell. And this is also not distinguish the live and dead cells. So these four methods are indirect methods of measurement. Turbidimetric method, electronic enumeration of bacteria, and based on the metabolic activity and dry weight of the cell. These are the methods for measurement of microbial growth. And these are the references. And thank you for your patient listening.